All right, so I left off last time talking about sort of the classic inflammatory response. And if you remember, we talked a lot about the way that that inflammatory response can be a really important physiologic response that we really need, but also that if it goes wrong, either in amount or in timing or something like that, then we have problems. And I told you about one specific example of that last time, which is septic shock. And septic shock was sort of the first thing that people really thought about with PRRs and with the inflammatory response process as a place where perhaps knowledge of this process might help us intervene in this medical issue. But there are a number of other ways that inflammation can be a bit of a problem. As I mentioned last time in response to a question, inflammatory cytokines are usually produced in really tightly controlled settings for really brief periods of time, for really brief parts of your body, things like that, for local inflammation. If you start having some of those cytokines produced for too long of a period of time, they can start acting on numerous physiologic systems. And so we've talked a little bit about how some of these inflammatory cytokines can impact blood vessels in local inflammation. If, however, blood vessels are being stimulated with inflammatory cytokines for really long periods of time, then we can start to see changes like general permeability changes, or even thrombus, which is sort of related to clotting in those vessels. We often can see these cytokines impacting muscle. If we start to see those cytokines going for a long period of time, we can see impacts on the heart, like low heart output. If we see some of those cytokines acting on other muscles and making the metabolic changes that are important for things like fever, we can start to see things like insulin resistance. And so you can see that if these cytokines are not regulated correctly, we can have a number of different changes to the body. One of the things that you are going to see over and over and over this semester is that immunologists love to talk about Goldilocks. That if something is too much, it's bad. And if something is too little, it's bad. It has to be just right. And so these are some of the examples of inflammation being bad if they are too much, if it's too much. And so what we are realizing is that a number of different disease states can be tied to chronic inflammation. So inflammation that's going on for too long a period of time. That could be because of an infection that is not resolved. So the infection that sort of your immune system doesn't ever get rid of. That could be because you have some reason why you are responding differently than others to the microbes in your body, particularly your GI microbes. So maybe if, say, you have a genetic change that changes how you respond to those, you might actually make an inflammatory response when you're not supposed to. We might also imagine having too much inflammation with hyperglycemia and obesity. There actually is some data on certain lipids turning on PRRs, as well as different types of autoimmune responses, allergies, organ transplants, and cancer. And all of those things, of course, can be impacted by one's genetics. Any of those could lead to excess inflammation over time, which could result in a lot of different tissue and metabolic changes. And as a result, all of the disease states that are listed at the bottom of this slide have been tied to chronic inflammation. And so things like obesity, type 2 diabetes, heart disease, atherosclerosis, organ failure, cancer, Alzheimer's, autoimmune disease, autoinflammatory disease, inflammatory bowel disease, allergy, asthma, organ transplants, etc., all have been tied to chronic inflammation. That's why I've mentioned in the past sort of the 9 of the top 10 causes of death worldwide all have a chronic inflammation component to them. And this is actually why in 2011, the Nobel Prize in Medicine was given for the discovery of PRRs. Because if we could figure out how to change this process or influence this process in some way, you can see that there are massive numbers of human disease states that we could potentially impact. And at this point, it's almost easier to say which diseases don't tie to inflammation as opposed to which diseases do. So everything that I have told you about thus far has been kind of this classic inflammatory response process. And it is usually thought of and is shown here as involving bacterial infection. And that will lead to some of these changes in the vasculature, that will lead to antimicrobial peptides and phagocytes like neutrophils coming in to phagocytose those bacteria, kill them, all the good stuff we saw last time. However, things get slightly different when we think about viral infection. And so we do think of a similar kind of parallel process being really important for viral infection, although there are a few unique differences. Here you can see the viral infectious cycle. What you can notice in either of these figures of the viral infection is that we start out with the virus interacting with the cell at the cell membrane, and then coming apart or uncoating in some way, so that eventually we have nucleic acid in the cell. And so here you can see the nucleic acid of HIV. Here you can see some general nucleic acid that's officially supposed to be polio. In any case, you see that there's this nucleic acid. And then a lot of enzymes in the cell will be involved in replicating that nucleic acid, translation, building some proteins, having that virus leave the cell. What you should realize here is that there are way fewer targets than there were in the case of bacterial infection. We don't have flagella. We don't have LPS. We don't have peptidoglycan as good targets here. Viruses are much simpler than that. They don't have as many of those kinds of biomolecules. In fact, in many cases when the virus gets into the cell, the only thing that really gets into the cell is the nucleic acid. And so we just probably are going to have to maybe detect the nucleic acid. It's not sort of as much of a different target. And the way we would deal with this virally infected cell is also different. We don't really want to phagocytose those viruses because their whole goal in life is to get inside of cells. And so if we phagocytose them, that's like we just did their job for them and got them into a cell. So phagocytosis is not always the best defense against viruses. If we look at this infected cell, there's actually not that much we can do for it. We can't go in and say, I like this RNA, and I like this RNA. Oh, this one's bad, I'll throw it away. And I like this. We can't sort at that level. Basically, once this cell is infected and has this nucleic acid mixed in with its own nucleic acid, the cell's a goner. There's nothing we can do for this cell anymore. What we really would like to do is stop it from being a factory to make new viruses, but we can't do much for this cell. And so the specific details of how we're going to recognize that virus and what is the best action to take are going to be a little bit different than you saw in the sort of classic bacterial case. I will tell you that some of those cytokines that I mentioned last time, IL-6, TNF, IL-1 beta, are definitely made in viral infection responses as well. I'm kind of oversimplifying this bacteria versus virus difference right now, but we can think about it in this way. And so our understanding of what goes on with viruses really started in 1957 with these two scientists called Isaacs and Lindemann. And Isaacs and Lindemann infected some cells with virus. I believe it was influenza virus. And they found that sometimes cells could make a substance that you could put on other cells and would block them from getting infected. It interfered with infection. There was this magic substance some cells made that interfered with other cells getting infected. I didn't know much of anything about it other than it was this magic interfering substance, so they called it interferon. We now know of 3 very major types of interferon, type 1, type 2, and type 3. And right now, kind of for the rest of our time today, honestly, for the rest of our time for a while of the semester, we're going to talk about type 1. And so type 1 interferon is most famously 2 types, interferon alpha and interferon beta. They are made really quickly after infection, so very, very rapidly when cells are infected with a virus, and they often decline by about 10 hours after infection. So when a cell is infected with a virus, it makes type 1 interferon almost immediately. There are other types, and you can see here, type 1 interferon, alpha and beta, can be made by most of our nucleated cells. So basically, you can imagine just about any cell of your body can make type 1 interferon almost immediately following viral infection. Type 2 interferon is called interferon gamma. Interferon gamma is part of the adaptive immune response, and we will talk about it then. There is also type 3 interferon called interferon lambda. It is made by epithelial cells, and it seems to actually be one of the key things that helps a barrier organ be a barrier organ, although there is far less known about some of its details. So we're only going to be really talking about type 1 here, which was the type that was described by Isaacs and Lindemann back in 1957. And so this is how type 1 interferon works. In a sort of big picture level, we're going to zoom in a little bit more. So what you can see here is that we can have a cell that becomes infected with a virus. And that cell might then become a factory for new viral progeny to be made, just to allow viral replication. When that cell is infected with the virus, it will start to make this cytokine, the type 1 interferon. That type 1 interferon is going to really importantly be made by this cell, transcribed and translated, but also secreted. And when it is secreted, it is going to act on an interferon receptor on a neighboring cell. And so you can see the importance of interferon is not for the cell who makes it, it's for that cell's friend, the neighboring cell that does not have a virus. And what this is going to actually do is this is going to change this uninfected cell and make that uninfected cell resistant to viral replication. So basically, that is going to turn that uninfected cell into something known as having an antiviral state, so it can block virus replication. So what you should realize is going on here is that we've got 2 cells that are important in this process. One of them gets infected with a virus. The virus gets in. Stuff happens. And that cell makes interferon. That interferon then acts on the interferon receptor. The official name of the interferon receptor is IFNAR, on this neighboring cell. And that makes this cell go into the antiviral state. So that's kind of big picture what goes on here. We can also go, zoom in a little bit more on some of the specific details. And so this also gives us a little more information on that antiviral state in the uninfected cell. So here you can see we've got signaling happening in our infected cell to result in the production of interferon. And then you can see that interferon acting on a receptor on a different cell and leading to another set of signaling. Type 1 interferon binding to its interferon receptor will actually make lots of changes in transcription in this uninfected cell and lead to the production of thousands of new transcripts and thus thousands of new proteins. So it's a huge array. All of these genes themselves are known as ISGs, or interferon-stimulated genes. So basically, this cell starts to make ISGs, and that's how it gets the antiviral state. One thing that all of these genes have in common is they all have the same promoter, or a very similar promoter, called the ISRE, or the interferon-stimulated response element. That allows for this transcription. We'll talk a little bit on the next slide about how exactly some of these ISGs are actually able to give you an antiviral state. Because right now I've told you they make antiviral magic, but I haven't really told you how. One other thing that just to be aware of is that the combination of ISGs that are made in any particular infection are probably important. So you might make ISG number 4, 52, and 312 when you get infected with flu. And you might make 7, 56, and 709 when you get infected with HIV. And those differences are probably important. But we're only now getting to the point where we can actually measure enough genes at the same time to really start understanding what's going on. So those sort of specific profiles are probably important, but we don't know a ton about them. So here are some particularly famous examples of how the antiviral state can work. And so you can see we've got our interferon protein at the top binding to the interferon receptor and leading to transcription of our ISGs. One of the most famous ISGs is known as PKR, otherwise known as protein kinase R. Protein kinase R has a really important job. Protein kinase R is able to lead to the phosphorylation of the protein shown here. And the reason why anyone cares is because this turns off translation in the cell. So if we turn on PKR, this makes our uninfected cell stop translation. If you think about it, that's a pretty good way to combat viral infection of the cell. The cell is, or the virus is trying to use the cell as a factory to make new virus particles. To do that, the cell needs to translate viral proteins. You turn off translation, you can't make new viruses. And so this cell now is no longer going to be able to serve as a kind of a factory for viruses. Another particularly famous one is called OAS, 2', 5' oligo-A synthetase, which leads to the production of something called RNase L. This is an enzyme that cuts up mRNA. So it just chops up mRNA. Again, you can imagine how that could be pretty useful in combating viral infection in a cell. If the virus is trying to use a cell as a factory, wants to do translation, before it does translation, it's got to have some mRNAs to get translated. If you just cut up all the mRNAs, then no more making virus proteins. So we're going to have this nice antiviral process. There are many, many other ISGs. You can see that some of them, again, inhibit transcription. Some of them also inhibit translation. One of my personal favorites is one called ISG15, which does a lot of things, but one of the things it does is it just basically puts a sticker on some random proteins, and then when they try to fit together to build a virus, they can't because they have this random thing on them. So there's a lot of really cool ways that all of these proteins are going to help the cell go into an antiviral state. However, there is also a massive problem here. So sometimes when students hear about this whole process, they say, okay, this is cool, but also this seems really dumb. So we have this whole program we can turn on in the cell to make sure that the cell cannot be infected with viruses. Why don't we just have that on all the time? Why do we ever not have it on? I'm telling you that we have this special thing. In fact, I told you a bit ago, this happens really rapidly within your cell and then stops by 10 hours. So we have this awesome process that's going to get turned on and stop our cells from being able to be infected by viruses. And you might wonder, well then, why stop it? And we've got to think a little bit about that piece too. Yep, Sydney. Yeah, this doesn't say we inhibit translation of just virus proteins. We just stop translation in the cell altogether. So yeah, we can't translate any virus proteins, but we can't translate any of the cell stuff either. And so if this cell wants to do any of its normal processes, it's out of luck. We're also going to cut up all the mRNAs. And it's not going to be just the virus's mRNAs. We're going to cut up all the mRNAs in the cell. It's good because we're going to get rid of the virus RNA, but it's bad because we're going to cut up all the mRNAs the cell needs. Many of these genes, while they're really useful in blocking viral infection of the cell, are also not that healthy for the cell to have on for a long period of time. The cell can survive with these products made for a short period of time to stop a virus that might be around, but these are really, really bad for the cell. And so interferon is going to induce these gene products that cause big problems for cells. Now, if we think about interferon and it being turned on, you could imagine that we can think about the cells with an interferon receptor. You'd want to have an interferon receptor on any cell that might get infected with a virus in your body, or whose neighbor might get infected. Basically, you want all your cells to have ears listening for that call from their neighbors. What kind of cells of your body can be infected by a virus? Almost. All except one. And the one that cannot be infected by a virus are red blood cells, because they have no nucleus, as you saw in lab 1. So basically, every cell of your body, if it has a nucleus, it has interferon receptors, which means every cell of our body can have its transcription messed up and its translation messed up by this process. This is one of the reasons why if you have a lot of interferon in your body, you feel bad, because your cells are suffering. You are going to have fever, you're going to have chills, you're going to have nausea, you're going to have malaise. And basically every viral infection leads to interferon production, which is why every viral infection basically leads to the same set of symptoms, the quote-unquote flu-like symptoms, which immunologists and virologists call acute viral infection symptoms, because they're just the things that interferon does whenever you make too much interferon. In addition, interferons can actually inhibit other arms of the immune system because you don't want to have kind of an immune response going on too long, too much. You would like to make sure that you sort of keep it under control so you don't start to have autoimmunity and cell damage. And so, in fact, there are some situations, 1918 flu is actually the most famous example of this. Most 1918 flu patients died, or one of the big reasons why they died is bacterial infection, because they had so much interferon it turned off the antibacterial response. And so then they ended up dying of bacterial infection. So there's also this kind of interplay where interferons can actually suppress some other aspects of your immune response as well. So hopefully, with what I have told you right now, you can kind of understand why immunologists are really excited about understanding pattern recognition receptors and how pattern recognition receptors respond to microbes, because we could intervene in so many different aspects of immune responses. And so now for the rest of the time today, I'm going to talk about some specific groups of pattern recognition receptors and their kind of important features and their important goals. So this is a table from your textbook talking about some different groups of pattern recognition receptors. And we're going to talk about some of those different groups. In talking about them, most immunologists think about these groups based on the first group that was ever described. So there's one group that was described first. We kind of understood how it worked, and then everybody just wants to make all the other ones kind of like match. So I want to tell you first about the first group that was described. And in past years, I have kept count as I've gone through the semester of how many Nobel Prize winning experiments I've talked about. I have not been keeping count thus far, so I can't tell you how many I've already told you. But this one, here's one, if nothing else. And this experiment and this gene actually have won 2 Nobel Prizes. So to start out with, I have to tell you a little bit about this gene and the first Nobel Prize, which is sort of not an immunology Nobel Prize that happened with this gene. So this gene was first described in Drosophila, and it was described by some German scientists, particularly one named Christiane Nilsson-Volhart, who were trying to understand developmental biology. She was trying to understand something called dorsoventral patterning. So basically how an embryo gets a front and a back, or a head and a tail. That was really what she wanted to understand. And she was making mutations in her flies and then looking to see if any of them messed up the embryo. So like the embryo had 2 heads or 2 tails or 2 fronts or 2 backs. And so that's what she was trying to do. And at one point, she made this mutation. People who work on Drosophila love to name their genes funny things. So she made this mutation. And the embryo suddenly lost dorsoventral patterning. So it was super messed up. And Christiane Nilsson-Volhart was German. Does anyone here speak any German? All right, you're going to have to help us with something, okay? It's okay. I've taught this a number of years. I know the answer to the question I'm going to ask, okay? But she looked at the microscope, and she said, "tall," when she looked in the microscope at these embryos. What's that mean?
It's funny, I actually forget what told me.
So in fact, I've heard a few different definitions of it. I've heard a few different ways of phrasing it. So I could also imagine that you just don't want to say what you think. Because I've heard definitions ranging from, "oh wow," to "oh shit," as being what it means if you're like, "tall." I don't speak German, so I don't know. But that's what she said when she saw the flies under the microscope. And so she named the gene Toll. And eventually, some other scientists, and she won the Nobel Prize for that. Eventually, some other scientists figured out a way to make some flies that were missing this gene, toll, but that only started missing it when they were older flies, when they had developed. So it didn't mess up their embryos. They had normal embryos. It was just when they were adults that they didn't have the gene. And when they did that, they got this. You may not be used to looking at Drosophila under an electron microscope. So what I will tell you is, see all this green here? That's all fungus covering this Drosophila. And so what they found is that if you had Drosophila adults and they did not have this gene, toll, they had no ability to respond to fungal infection, and they died of massive fungal infection colonization. And this was on the cover of the journal that first described this. Like, this is a very famous picture to immunologists. So it was realized that toll was actually the first of the PRRs that was really described, that it could respond to a microbial product and lead to this inflammatory response. We now know of a whole family of receptors that have the same kind of structure and that act very similarly. Toll is actually the name of the gene in Drosophila. In all the other organisms, these are known as toll-like receptors because they're like toll. They all have this domain outside of the cell, or this domain that can bind to ligand called a leucine-rich repeat, as well as other parts that are involved in signal transduction. In fact, toll-like receptors were really hard to study for a long time because of those leucine-rich repeats. It turns out that in terms of biochemistry, they are super, super, super sticky, and nobody could purify these proteins and do some of the experiments everybody, like the field, wanted for years because these are so sticky. We now know that toll-like receptors can be divided into two groups. One group lives on the plasma membrane. And so you can see a whole bunch of toll-like receptors that live on the plasma membrane in this figure from your textbook. In fact, you can also see that they usually dimerize, whatever. And if you look at these plasma membrane toll-like receptors, or the extracellular TLRs, you can notice some things about what they bind. So you'll notice that TLR4 binds gram-negative bacteria. TLR2 kind of is bacteria. TLR5 is flagellin. So you kind of see a lot of bacteria stuff as the ligands for these cell surface TLRs. Alternatively, there are a group of TLRs that are found inside of a compartment in cells known as the endosome or the lysosome. And so it's basically a whole separate group. And what you can notice is that the TLRs in the endosome are largely binding to products that come from viruses. So we've got these two groups that can be distinguished both based on their location in the cell and by what type of microbe they tend to recognize. It turns out that if you, again, look at these and this distinction, you can also see some other distinctions in their signaling. So those cell surface toll-like receptors, as I mentioned, are mostly binding to ligands from bacteria. When those cell surface TLRs bind to the ligand from bacteria, which as is shown on the signaling pathway to the left, they lead to some signaling that turns on a specific transcription factor. You don't need to worry about all the steps in between. I just want you to know the transcription factor they're turning on. And they turn on a transcription factor called NF-kappa B. This is my excellent bacteria, because I'm really good at art. You will learn by the semester how good at art I am. We've got our bacteria. We've got our toll-like receptor. It is binding that bacteria outside of the cell because the receptor is on the surface of the cell. We're getting signaling in the cell. The cell is making NF-kappa B transcription factor, or is activating NF-kappa B transcription factor. And it turns out when you make NF-kappa B, NF-kappa B specifically tells the cell, hey cell, it's time to make IL-1. It's time to make TNF. It's time to make IL-6. It's time to make chemokines. These are all of the products that we saw in that classic bacterial response that I told you about last time. So they are all specifically made if you turn on NF-kappa B. And so all of the sort of classic parts of inflammation that we talked about before all happen because NF-kappa B got turned on. There are also some other things that are really important for turning on adaptive immunity that we'll talk about much later in the semester. And so this kind of works out. We have a receptor that's in the right location to find bacteria. It only can bind bacterial products. And the final result is you make a response that's good for killing bacteria. Seems like a nice plan. Alternatively, the TLRs that live in the endosome turn on a completely different type of signaling. And when they turn on their signaling, they turn on some transcription factors called Irfs. IRF is interferon regulatory factor. Based on that name, what do you think IRF makes? Yeah, interferon. It regulates it. And so, in fact, if we have a toll-like receptor inside of the cell, it's in the perfect place to recognize parts of viruses. And it will make a different transcription factor. This is also going to be a type of PRR that only recognizes virus stuff. Then it's going to make a transcription factor, IRF, that tells the cell, hey, let's do this antiviral response instead of doing that antibacterial response. And so it gives us sort of this nice tailor-made response. And so here you can see, if we turn on IRFs, alternatively we get the expression of type 1 interferon, and we get the antiviral state. And so what you can see is the type of receptor that senses the pathogen, as well as the location of that sensing, will dictate what type of response we have and help us make an appropriate type of response. Since the toll-like receptors were described, there have been a number of other groups of pattern recognition receptors that have been described. And so I'm going to kind of briefly go through those other types now. So this slide mentions something called the CLRs, or the seep-type lectin receptors. I'm not really going to talk about them. My knowledge of them is they're important for fungi. But we also have a group called the RLRs. So TLR stood for toll-like receptor, because the first one of them ever described was toll. RLR stands for RIG-I-like receptor, because the first one of them described was RIG-I. So I'm going to show you a couple of things about the RIG-I-like receptors that are important. And hopefully you'll see kind of some general themes that are emerging. The RIG-I-like receptors are found in the cytoplasm of cells. and they bind to nucleic acid, specifically viral nucleic acid. So we see cytoplasm location, we see binding of nucleic acid. Just based on that, what transcription factor would you guess, even if you didn't see it on the slide, what transcription factor would you guess that RLRs turned on? Yeah, Sebastian? Irfs, and they do. So the RLRs, again, they're responding to viruses. They're in the location to look for viruses, so they turn on the transcription factor that makes the antiviral response, the IRF. That's honestly like pretty much what's important about the RLRs. So the next big form of pattern recognition receptor is one that's called the NLR. So remember the TLRs were toll-like receptors. The CLRs are C-type lectin receptors. The RLRs, RIG-I-like receptors. You're kind of getting how this naming convention works. So the NLRs are the nucleotide binding domain leucine-rich repeat containing proteins, which looks nothing like the naming of the other ones. Super wacky. The actual reasoning here is there was an N protein called NOD, and we said, oh, these are the NOD-like receptors. We're going to call them the NLRs, NOD-like receptors. And then people did some more experiments, and they realized that NOD was nothing like the rest of them. And so none of the rest of them were actually NOD-like at all. So NOD-like receptor was a really bad name. And so then they said, well, we want to keep NLR. And so let's figure out a name that we can pretend works for, is what NLR stood for. And so this is what they came up with. It does, in fact, describe some important features of the structure of these proteins. So that's pretty cool. You can see some examples of NLRs at the bottom of this slide. And you can also see two other proteins at the top that are not NLRs, but that have some pretty similar structures. So you can see that, in fact, all of these have something called an NBD, or nucleotide binding domain. That's a fancy name for they use ATP. And they all have the same domain to use ATP. You can see the NLRs have this other domain called a leucine-rich repeat. That's the same thing that we have in TLRs. It's actually in a lot of pattern recognition receptors. And that protein at the top also has NBD and LRR. This protein in the middle has NBD and something that looks an awful lot like LRR called a WD40 domain. So you can see it's a pretty similar kind of structure. And then they all have basically other at the N-terminus. And there's some similarities between the others, too. So you might wonder why the heck I'm showing you these other random proteins. Well, NBS LRR proteins, the ones at the top, are basically the entire components of plant immune systems. And so what you can notice here is some convergent evolution that using kind of the same types of proteins, plants and animals have come up with very similar strategies for their immune responses. The other protein that's on here is called APAF1. You actually may have learned you have heard of APAF1 before. You just might not remember it or might not know it. APAF1 comes up in Bio 250. And so I want to remind you of APAF1 for a second, because you can see APAF1 is pretty similar to these NLR proteins. So when you were in Bio 250, at one point you learned about activating caspase 9 and caspase 3. And this was in the process of learning about apoptosis. And when you learned about apoptosis, you learned about this structure that gets made in the cell. Any of you guys remember that structure? Dr. Dunaway, the official name of that structure is called the apoptosome. Dr. Dunaway's name for that structure is the Pinwheel of Death. Do we remember the Pinwheel of Death? So that one usually rings bells, even if the apoptosome doesn't. The apoptosome, the pinwheel of death, is made of APAF1. It's APAF1 coming together in a cluster, in a whole bunch of copies of itself, and turning on caspases so that we get apoptosis. And so you can see APAF1 has a very, very similar structure to NLRs. And you will care, I promise. This is not random fact time, although I do love random fact time. One thing that is a little bit tricky with the NLRs is that different NLRs can have different functions. Some of that is based on what this other domain is. When I think about NLRs, there are some that have names like NLRP1, NLRP3. They get that P in the name because they have the other domain that starts with P, a pyrin. So NLRP1 is the first one described that had the pyrin. NLRP2 was the second one. NLRP3 was the third one. There are some that are called NLRC4 or NLRC1 or whatever. It's because they have this card domain. Others can have some other types of domains. And so some of the functions will vary a little bit based on that. But if we think broadly, there are two big functions that NLRs have. One of those functions is that they are able to induce cytokine production. Some of them bind to bacterial products. Which transcription factor would you bet the ones that bind bacterial products activate? Sydney. NF-kappa B. Some of them bind to viral products. Which would you predict? Which transcription factor would you produce? They predict they would produce. Yeah, Aaron. Irfs. So we can kind of see sort of the same broad patterns of cytokine production that we saw with the other PRRs. NLRs, however, have this other bonus function, however. I said however twice in that sentence. That's weird. And that is in the production of a protein complex that looks like this. It contains multiple copies of our NLR protein, which, weirdly enough, looks like APAF1. It contains multiple copies of this other protein called ASC, which, weirdly enough, looks like cytochrome C. And it contains multiple copies of a caspase called caspase 1, which looks kind of like caspase 9. And they come together to make this structure. So, does this structure look like anything you have seen before? Yeah? It kind of looks like a pinwheel of death. Now, the pinwheel of death is called the apoptosome. It's a protein complex that's made during apoptosis. This protein complex is not made during apoptosis. This protein complex is made during inflammation. So we call it the inflammasome. But structurally, it's almost identical to the apoptosome. With the apoptosome, you were turning on a caspase and making it so that caspase could cut stuff. And when the caspase cut stuff, the cell went into apoptosis. Here, the caspase is going to cut stuff, but that cutting is going to actually lead to cytokines being produced. And those cytokines, IL-1 beta and IL-18, require this caspase. So every caspase in the world is not involved in apoptosis. There are apoptotic and non-apoptotic caspases. This is a non-apoptotic caspase, and it partially turns on these cytokines. So you can see that in this figure from your textbook as well, our NLRs can come together into a complex along with ASC and caspase 1 to give us active caspase 1, which can give us these cytokines and allow them to be released from the cell. In addition, there is one other thing that the inflammasome can do, and that is shown both on this slide and the slide before. But I need to define it a little bit. So this is something known as pyroptosis. And you can see pyroptosis listed here. And so you guys learned in Bio 250 about apoptosis, which you learned as programmed cell death. And you also learned about necrosis. So it turns out that's a little bit of an oversimplification of cell death. Programmed cell death means the cell tried to die. There was signaling in the cell dying. The cell kind of was committing suicide. Apoptosis is the most famous way the cell can commit suicide. It's not the only way. Cells can actually, necrosis you learned about is kind of like the cell just blows up by accident. And apoptosis was like the cell planned it. There are more other ways the cells can plan to die than just apoptosis. Actually, one of them is netosis, the thing we saw with neutrophil nets. But the other one that we need to talk about here is apoptosis. So this is a little bit of a description of the difference between apoptosis and necrosis. You can see there's an awful lot of signaling with some caspases for apoptosis. With necrosis, we don't really see the same kind of planned signaling process. And in the end, the cell blows up and releases all of its contents. The end of apoptosis, the cell makes these little membrane bodies. It basically folds up little bits of membrane around all the parts of the cell and makes little packets. Whereas in necrosis, the cell goes, okay? So there is this other form of cell death called pyroptosis. Pyroptosis is programmed necrosis. So in this case, there is cell signaling, specifically cell signaling using caspase 1, where the cell then swells up and explodes and does basically what you learned about previously as necrosis. So we have membrane swelling, membrane rupture. We release all the intracellular contents. We don't make the nice little bubbles. We don't make the nice little apoptotic bodies or blebs. We explode that cell. And as an immunologist, I think about these two forms of cell death. Again, both programmed cell death, both the cell is dying on purpose, as having some particularly important differences. So when I think of apoptosis, I think of apoptosis as very quiet cell death, where the cell is going to die and not tell its neighbors and not make a problem for anyone else and not bother anybody. That's apoptosis. When a cell undergoes pyroptosis, what does pyro mean? Yeah, fire. Pyroptosis is fiery cell death. Pyroptosis is when the cell is dying and letting everybody around know about it. That cell is just making a huge fuss while it dies. And what you can imagine is that that can actually alert neighboring cells to the fact that there might be a problem. This could potentially do things like release those damage-associated molecular patterns that came up in class last time, and be basically, I think of them as either immunologically silent death or immunologically noisy death. And whether or not the cell is going to disturb its neighbors and try to tell its neighbors to become activated. Pyroptosis is also induced by the inflammasome. And so NLRs, in addition to leading to cytokine production, will also induce pyroptosis, this very loud cell death. And you can see it's kind of interesting that the cell uses two very similarly structured proteins, this NLR for inflammation and pyroptosis, versus APAF1 for the silent death, apoptosis. So you can see kind of the parallel there. No problem. So this figure from your textbook also shows one other kind of thing about NLRs. So NLR P3, and this is actually just one NLR on this slide. And this shows you all of the things that have been indicated as possible triggers of NLR P3. So you can see that this includes ATP, cholesterol, uric acid, glucose, amyloid beta, hyaluronic acid, alum salt, asbestos, silica, UV radiation, skin allergens, alloys, bacterial proteins, virus proteins, fungus proteins, parasite proteins. Any of you who are biochem majors are going to look at this and be like, there is no one receptor that binds all those things. Those things are not the same. You cannot have one receptor that can match them all. And that's actually something the field is still working on. There was actually a paper that was published yesterday that I saw on Twitter that's purported to have an answer, but I only saw the title, I didn't read it yet. Actually, I've read other things, I have a hint of what the answer is, but so we don't really know, but this also can show you just how broadly these NLRs could be important in a lot of different disease states. In fact, if you look at the inflammasome and look to see either is a disease linked to mutations in the inflammasome, is the disease cured when you block the inflammasome, or is the disease linked to the inflammasome in an animal model? So here the genetic part is in human disease. You can see a list of some of the human diseases linked to the inflammasome, including inflammatory bowel disease, type 2 diabetes, hypertension, Alzheimer's, atherosclerosis, dermatitis, type 1 diabetes, multiple sclerosis, lupus, vitiligo, Addison's disease, celiac disease, rheumatoid arthritis. This is actually a super out-of-date slide, as well as some weird diseases called autoinflammatory diseases, where you have excess inflammation for strange reasons. So one of those, I think, is FCAS, but I should have looked this up before class today. One of them is actually extreme sensitivity to temperature, so that you'll break out in hives if you take a hot shower. Yeah?
Actually, I had a class in high school, but it wasn't a hot shower.
Yeah.
Below a certain temperature.
Yep. Yeah. And that's because she has that is linked to mutations in PRRs, so that you have inflammation at the wrong time. And so all of these things seem to be related to this process, which is why people spend so much time studying the inflammasome. The other group of PRRs that I want to mention really briefly is the last group that your textbook talks about. And these are known as the ALRs, which are shown here at the bottom. So you can actually look at some of our other receptors. You can see the plasma membrane ones, and those being the bacterial ones. You can see the cytosol ones, perhaps having something about viruses. You can kind of see some of these tie-ins. The ALRs, you should note, are found in the cytoplasm and also, really interestingly, in the nucleus, because some viruses try to take advantage of the nucleus and hang out there. I also like the ALRs because that's what my lab works on. In humans, there are 4 ALRs. They all have one part in common, the pyrin domain. It's the same pyrin domain that the NLRs have. And another part that binds to DNA. And so all of these proteins are going to bind DNA to induce a response. And they will actually go through a common, some common signaling. And they have 2 different outcomes they can lead to. One of those outcomes is IRF induction and the general antiviral state. So typically we'll see viral DNA getting sensed either in the cytoplasm or in the nucleus. And that turns on IRF, and we get interferon production, just like we should for any cytoplasmic viral sensor. Alternatively, these receptors can also form inflammasomes and lead to pyroptosis and that process. And so these AIM-like receptors can do that. So that's kind of this general idea. Again, you can see why they might be interesting to think a lot about. Your textbook also has one other thing that they put in next to the ALRs. And this makes me snicker, because they put in this other protein that is not an ALR. and they just kind of lump it in by the ALRs. It's a protein that's called CGAS. My lab also works on CGAS, so the fact that the textbook just kind of throws them all together cracks me up a little bit. So CGAS is an enzyme. CGAS is called Cyclic Gmp AMP synthase. It makes a little small molecule, kind of like cyclic AMP that you learn about in other types of signaling. It makes this little bitty molecule, CGAMP. You can see it right there. CGAS is CGAMP synthetase. CGAMP can lead to signaling. It's actually very similar to some of the ALR signaling, and eventually the production of Irfs and interferon beta, the type 1 interferons. So it's the same general process. In fact, a big part of my research is on ways that ALRs and CGAS are similar and different. So again, I think it's funny that the textbook lumps them together. And you will notice that in your textbook, CGAS is kind of its own little orphany thing. Everybody else gets to be in a family. The LRs and the NLRs and the RLRs and things like that. And then there's CGAS. CGAS is actually related to a couple of other immune proteins, though they are more distantly related. And that was the story until this summer. This summer, there was publication of an entire new family of CGAS-like receptors. So basically, CGAS's family was found this summer. So now there is a CGAS-like receptor family that Philip Kranzuch and his lab just described this year. And so we are still, I think, probably only scratching the surface of PRRs. And these have some ways that they are similar to CGAS, some ways that they are different. And so exactly the rest of the details of this CGAS-like receptor family remains to be determined. So what I have told you about here are a lot of aspects of how innate immunity works. Some parts of the antiviral state even start to fall into intrinsic and can get a little bit barrier responsey. Don't worry about, I will never ask you where if the antiviral state is here or here, because it's here. But we've really kind of talked through these innate immune processes. What happens in the first, say, 96 hours after infection? On Wednesday, we're going to start talking about some aspects of adaptive immunity. And we are going to start to see aspects of adaptive immune responses and how some old classical ones were discovered and how they work and move into adaptive immunity. And so I will see you Wednesday when we start talking about that.